Welcome everyone to Lecture 1 of the first section of ZOO3649 for 2020. I'm Professor Moodley, as you know, and we're going to start this section with the first two lectures are basically getting you up to the modern day. All right, everything we discussed in second year until now has been about how things were done in the old days. All the different DNA markers, all the ways to visualize recombinant DNA. In the modern days, if you ever do postgrad, the only kind of DNA data you're going to be seeing are DNA sequences, okay? And there are two kinds of DNA sequences that we deal with in the modern day, right? We're still using Sanger DNA sequencing. This is what we're going to be talking about today. But we're also using new technologies called next generation sequencing. And this is what we're going to be talking about in the next couple of lectures. So, the first generation, as we learned in 2544, the first generation of DNA sequencing included two kinds of methods. You remember, the first method was the Maxim-Gilbert based destruction method, and the second method of DNA sequencing, only a few discovered or figured out only a few years after the Maxim-Gilbert method was Sanger's method, okay, and that's the chain termination method. So, if you want more details about either of these methods, including the Sanger method, please go to your ZOO2544 course guide, look at the lecture about visualizing recombinant DNA. Okay, and there you will get all the details about the Sanger method. We're not going to be concerned about the Sanger method too much in this module. We're going to be concerned mainly about next generation sequencing. But I'm giving you this because it could be, in the modern day, if you are doing a postgraduate work, you will see Sanger sequences and you will have to be able to deal with them, right? And this is why we're dealing only with DNA sequencing in third year. Just briefly go through the Sanger method. Now, firstly, the locus. What is the locus? The locus is the piece of DNA or the place in the genome that you want to find out the sequence of. Okay, you want to find out the sequences, and the sequences are made up of letters, right? 4 letters to be specific, and you know them: A, C, G, and T, right? But they're in different sequence, and how they're arranged, that sequence of the DNA, is very important to know, because you know from the central dogma that sequence is what codes for messenger RNA and what codes for proteins. So that DNA sequence is actually got all the message, the code for life itself, for making an organism from an embryo, from nothing to life. Okay? So knowing a DNA sequence is probably one of the most important things in molecular biology. Knowing what is the DNA sequence of whatever you are interested in. Whether it's a virus like COVID or HIV, or a bacteria like TB, or a parasite like malaria, or a rhino, or an elephant, or a tree. It's all about DNA sequencing. Okay? So, first you need to find the place of the genome that you're interested in, a particular part of the genome, and remember, Sanger sequencing can't sequence the whole genome. Can't. It's the first generation sequencing. Okay? So this is sequencing only tiny parts of the genome. That's a big disadvantage of Sanger sequencing compared to the ones we're going to be talking about afterwards. So you sequence a piece of the DNA, a locus, one locus or gene, or whatever that you're interested in, a place of the genome that you're interested in. You usually get a lot of copies, make a lot of copies of that locus of interest. You can do it through cloning and amplifying through cloning, or you can do it through PCR. That's the modern way to do it, by PCR. You make a lot of copies of it, so now you have a lot of copies of it, which are called what? PCR products, right? And you sequence these PCR products using the Sanger method, all right? And you use the same primers as the PCR primers as your Sanger chain terminating primers, the ones that start the chain termination process. Okay, so use the very same primers that you did to amplify the gene with PCR. You do a second sequencing PCR then, and using the same primers to do the primer extension and the base destruction as you go along. So, and therefore now, because you use the same PCR primers for maybe you have taken COVID-19 viral isolates from 10 patients, no? And you want to see, do all of these patients, are they all infected with the same COVID? How do you do it? You get your COVID, you isolate it, you amplify with PCR, you then sequence it with the Sanger method, and then what? You have the line of DNA sequence for one. Then you have a line of DNA sequence for the other one, and all 10 of them. Then what do? You know, because you use the same PCR primers for all 10, right? You amplified the very same part of the genome, right? It is a homologous part of the genome. This is what we're going to be talking about a lot later on in the module, homology, okay? That part of the genome in all 10 of those individuals have a common ancestor. Therefore, you can line them all up with each other. That is called a DNA sequence alignment. It looks like this, what is on your screen right now. All right? Do you see that? You watch my mouse here. G, T, C, A, C, A, A, G, A, A, G, C, G, T, T, G, G, and so on. That's the sequence. This is between, this could easily be the sequence of COVID, right? These are the base pairs in somewhere in the middle, around 3,000 base pairs into the alignment, okay? And you see here, each line is what? Each line is one individual, all right? It could be you, it could be your DNA there on line 1. It could be my DNA on line 2. And line 3 could be somebody else's DNA. Or let's just say we were going back to our old example, we were looking at COVID-19 patients. The first line could be the COVID of the patient 1. The second line could be the COVID of the patient 2. Now let's just say these were all patients. What can you see immediately from this DNA alignment? Who's clever out there? You can see immediately from this DNA alignment that these sequences, there are parts of them that are pretty much the same. No? Look at here, where my mouse is. Look at these ones here. Look, everybody, no matter which individual it is, 1, 2, 10, 20, they all have this part here. This AAAG. You see? Maybe this guy doesn't have it. But you can see they're all pretty similar. They all have this T. Yeah? They all have a lot of pieces of the DNA. Like this C here. They all have it. So you can see that they have some similarity between them. Let's just say they are COVID isolates. You can see they have similarity. But the most important thing you can see is what? That they're not identical to each other. Okay? They're not identical to each other. They are different. There are maybe even one mutation change from a T to a C or something. Yeah? And they're different. So, if you can know the DNA sequence, now say again, this is COVID-19, and there are several strains here of COVID-19 that we're looking at. So what can we tell? We can tell among all these patients there are, and we can count up how many mutational changes there are in this alignment. And we can work out the rate of mutation. Remember, mutation, we're going to get to it, but it's one of the 4 forces of evolution. And the way to measure it is through the rate of mutation. You need to measure how frequently does a mutation occur. That is the rate of mutation. And you can figure that out from this DNA sequence alignment. You can figure, so imagine you knew the rate of mutation of COVID. You can predict how infectious it's going to be. You can predict how it's going to evolve. You can predict whether it's going to become virulent in the future. You need to know the DNA sequence. do I make myself clear why is it important to know a DNA sequence? Not just for COVID. Say these were now not COVID. These sequences were different rhinos. And we wanted to manage the population of rhinos. We want to see all these mutation changes because it tells you what? It tells you there's a lot of genetic diversity there. A lot of genetic variation is among these rhinos here that we've sequenced. Okay? So knowing the sequence is very important for so many things. For so many things. Okay? That is why we're learning about DNA sequencing. All right. So each change here, each color change here, for example, many in this position, this locus, at many individuals have a C, which is in blue, but many also have a G. Some have an A. Okay, so many have a C, some have a G, one has an A couple of them have T's. So at that position, there's been mutations to change it from a C to a T, for example. So those are called substitutions, okay? When you substitute one for the other. And indel, okay, it can either be an insertion or a deletion. They change the reading frame. But indels and substitutions are collectively known as, well, you can call them mutations. But there are many other kinds of mutations, as you know from 2544. So the right way to call these things is polymorphisms. And they're polymorphisms of one nucleotide. You either substitute it, you insert it, or you delete it. But only one nucleotide. So that's why we call them single nucleotide polymorphisms. Okay? And polymorphism is what? Poly means many, as you know, and morphism means shapes, right? So many shapes. That means many variants. Okay, that's the way of saying variation. Okay, the mutation from a C to a T, or a T to a G, or a G to a C. Okay, that's if you have many different bases in an alignment like this, you have a lot of polymorphism. So polymorphism is another way of saying variation. Okay, so you can even say single nucleotide variants. Okay, polymorphism is just a way of saying variation. All right, so what are the advantages of Sanger sequences? They are highly reproducible. Okay, they are much more reliable per base pair than any other kind of sequencing. They're very informative. As I've shown you from the last slide, imagine what you can tell from lining up a few DNA sequences. You can tell a lot. So they're very informative. Okay, and then you can tell also the lines of descent from DNA sequences. Okay, you can use the DNA sequences and reconstruct what we call a phylogenetic tree and figure out which of those DNA sequences is mostly closely related to whom in that alignment. Who's closely related and who's not closely related. We make a tree to figure out that structure. We're going to be talking about that in the section 4, DNA structure. But what are the disadvantages of Sanger sequencing? It is expensive. For the amount of base pairs compared to next generation sequencing, Sanger sequencing is very expensive. You need to know something about the locus that you want to amplify, right? Because you need primers to do the PCR to amplify the region before you can sequence it. So that means you need to know the sequence of the primers. If you don't know the sequence of the primers that amplify a particular region, you can't sequence anything using this method. Okay? So that is a disadvantage. You need to know something already about the DNA sequence in order to sequence it. I know, that sounds a little bit catch-22, but that is a major disadvantage of Sanger sequencing. And of course, the amount of variation or mutations, or polymorphism as we call it here, the amount of polymorphism depends on what? The rate of mutation. Okay? The rate at which bases change, form, morph from C to G, or G to A, or A to T. That is determined by the mutation rate. Okay? It gives you unphased data, Sanger sequencing. It gives you unphased data because you're only able to sequence a haploid. Okay? If you sequence a diploid locus, so haploid locus means it's only in one copy, right? Like mitochondrial DNA, or bacterial DNA, or viral DNA. But in diploids like us, we get a copy from mum and a copy from dad. So now you've used PCR to amplify a gene. The same primers will amplify the copy that mum gave to you. They will also amplify the copy that dad gave to you because it's the same locus. It has this PCR primers bind to both. So then you do a Sanger sequencing on that. What will you see? You will see mum's sequence on top of dad's sequence. And if they are very different, you will not be able to tell them apart from each other. Okay? So you will not be able to tell what sequence came from mum and what sequence came from dad. That's why we call it unphased. You can't tell what the parents were. You know this already from 2544, phased and unphased genetic data. So then what does that mean? It means we will have to take the Sanger sequences that we've amplified from diploid, and we'll have to do cloning. We'll have to clone them into a vector, and then we will have to purify each vector and sequence the plasma, the DNA that we've cloned, right? And in that way, we will pick up the two alleles from mum and dad. So Sanger sequencing does not give you phased data. You just have to clone it to phase it, okay? But you also, we did go through that in 2544 last year. Now it brings us to next generation sequencing. Let me tell you very clearly, next generation sequencing has created a revolution in not just medicine or biology or molecular biology or genetics. No. Those revolutions in those fields means that there's a revolution for humanity in general. Okay? We all benefit from NGS. Why? Because now we have genomes of data, not tiny little loci that we sequence through Sanger sequencing. No, we have the whole genome. Every locus is sequenced. And with all that information, it has led to this revolution in molecular, in biology. And we are biology, okay? Which means that this revolution is going to revolutionize our lives as well. It's going to make us better able to deal with pathogens. It's going to make us better able to conserve our biodiversity. It's going to be make us much better able to deal with the challenges that are coming in the future. Okay? And we've got big challenges coming in the future. So, this NGS brings huge amounts of data in a very short space of time. When I say data, what am I talking about? I'm talking about DNA sequences. Okay? You're not talking about a few base pairs like in Sanger. You're talking about millions, billions of base pairs sequenced overnight. Now, that is a huge amount of DNA sequence data. Okay? Now we have more data to answer the questions we had 20 years ago. We couldn't answer it maybe with little loci. But now we can look back and say, oh, we couldn't solve it with the Sanger sequencing. Now let's try it with NGS. And that is leading to some incredible biological discoveries. And some of those discoveries we are going to be talking about in this module. Okay? So stay tuned for that one. Now, that is the point here. The new data provide more insights and allows us to get to the bottom of things and dig up, formulate new questions, develop new questions and new hypotheses that we can test further. All right? And so the variation of the entire genome is available to us. Okay? And I always tell you guys that the genotype is your genome, right? How that maps onto the phenotype is determined by what? Phenotype is what? Is what you see. This is my phenotype, right? That's the phenotype. My genes, my genome is the genotype. How does the genotype map onto the phenotype? You know that you have to add, to genotype you have to add environment, right? You mix those together and you get the phenotype. So, knowing the entire genotype, that means now with NGS we know not just the genotype of one locus or another, we know the entire genotype, the whole genome. And we are able now better to map that onto phenotype. And by doing that, we become much more aware of what is environmentally driving phenotypic change. So huge progress thanks to NGS. Okay, there are several kinds of marker that NGS has given to us, this revolution. Okay, and the markers are not. I mean, you'd think, well, what do you mean there are many markers? Surely it's just the whole genome. The whole genome sequence is one of the markers we're dealing with. But that's kind of expensive to do, sequence an entire genome. You can sequence parts, big parts, not just like Sanger, but big parts of the genome. Yeah, and still get enough data without sequencing the whole genome. Okay, so there are different kinds of ways. So the cheaper ways of getting genome NGS data, the cheaper ways is to use what we call reduced genome representation ways. Okay, and that is basically those are markers that we use to genotype SNPs. We want to find out what are the SNPs, the mutations across the genome. Okay, but we don't have enough money to sequence the whole genome. So what do we need to do? We say, okay, fine. Well, we don't have money to sequence the whole genome. We can sequence maybe 1/10 of it, or 1/5 of it, 1/20 of it, right? So how do we do that? How do we do that? That is how what we call reduced genome representation. They're cheaper methods, but they still get a lot of data because they're using NGS the NGS technology. All right, so you can have RAD-seq, okay, and you can have GBS, genotype by sequencing. Both of these methods are pretty similar. They need restriction cut sites, which you then sequence. But there's also whole genome sequencing, which is the sequence, the entire genome, all the sequence in the entire genome. That is the ultimate genetic marker, okay? Because you don't need anything else and you got the whole genome. All right, so that's the ultimate genetic marker. Exome sequencing. What is an exome? Exome is the exons. All right, the exons, the part of the genome that is expressed. So as you know, it's only about 1% of the genome, right? But that's still millions of base pairs. Remember, the genome is 3 billion base pairs. 1% will be still hundreds of millions of base pairs. So exome sequencing is NGS because it sequences a lot of data, but it's only sequencing the genes that are expressed. Okay? The exons. The transcriptome sequencing, which is RNA-seq, is also not sequencing the whole genome. It's only sequencing what? The messenger RNA transcripts. So what do you need to do? You know this from 2544. You need to take a sample from a tissue, extract what? RNA, not DNA, and change it to DNA using what? Reverse transcriptase, and then what? Sequence it using NGS, right? So that way you get the sequence of every messenger RNA that is being expressed in that cell. It is a very, very, very powerful. RNA-seq is one of the most powerful tools in NGS. RNA-seq is the way you get to the bottom of diseases. You figure out what's going on in cancer cells. You figure out what's going on in HIV cells because you figure out what mRNA are those cells producing. And then you can figure out, well, how do I design a system to knock out that mRNA production, right? If you don't know the mRNA is being produced by COVID-19, how do you know that you need to get rid of it? That is because that is how RNA-seq enables you to figure these things out. What is being expressed? Not all the genes are being expressed in all the tissues all the time, right? So RNA-seq, you use different tissues and you figure out the expression profiles of each tissue. Which genes are being turned on, which genes are being turned off. Can you imagine how important that is to know? And then lastly, we have Amplicon sequencing. So Amplicon is basically, it's a PCR product. Amplicon is what happens when you do a PCR on a small piece of DNA, small lockers, not the whole exome, not the whole transcriptome, not the whole genome. Like what you would do if you wanted to Sanger sequence it, right? A tiny little lockers. But you want to sequence this for hundreds, if not thousands of individuals, the same locus. So what do you have to do? You get DNA from those hundreds or thousands of individuals, you extract each one, and then you do a PCR on each one using the same primers, right? The same 2 primers for all the PCRs. And then what do you get? You get an Amplicon, you get a PCR product for each sample, and then you put them all together and you send that off and say, okay, sequence everything that got amplified, right? You give each sample a barcode, and so when it gets sequenced and it comes back, you can tell, barcode blah 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 was sample 1. Barcode blah blah blah, which is a sequence, is sample 2, and so on and so forth. Okay? So actually, instead of doing them one by one with Sanger sequencing, which would be very expensive, you put them all together, all the Amplicons from all the individuals, give each one a unique barcode, and then send it for NGS, and it sequences everything at once. Okay? So that's Amplicon sequencing. I want to deal a little bit more with RAD-seq and GBS in this lecture, and then we'll finish off the lecture, and we will continue with whole genome, the big one, the whole genome sequencing in the next lecture. But let's quickly go through RAD-seq. You do know this from 2544, okay? But we're going to go through it again. So this method, and as well as GBS, the next method, both these reduced representation methods relies on what? Restriction of the genome first, okay? We don't want to sequence the whole genome. We want to sequence a small piece of the genome. But already, a small piece of the genome is a lot of sequence compared to Sanger sequencing, right? So what do we do first? We cut the DNA cut the genome with restriction enzymes, okay? And a restriction enzyme will cut it into millions of little pieces. How does a restricted, how do you visualize a restricted DNA? How do you know? Now you have a tube with DNA. You add a restriction enzyme to it. How do you know that the enzyme has restricted the DNA? You run a gel. So here is a gel. Here is a gel. And you've learned about gels. I taught you about gels in 2544. We asked you exam questions about gels in 2544. So now, don't come and look at this gel and be confused. What do we have in this gel here? You put the DNA here, right? You applied an electric charge between this part of the gel and that part of the gel. Which way did the DNA run? It went to the positive side here, because DNA is what? Negatively charged. And you also know that the big pieces were slow going through the gel, right? And the small pieces were fast going through the gel. So now, here's the positive side, where all the DNA was busy running. They started here. They ran here. So, where are the small pieces on this picture? Here, right? Down here are all the small pieces of DNA. Here, in the middle, are all the medium-sized pieces of DNA. And here are all the big-sized pieces of DNA up the top here. Okay? So now, how do we know that our DNA is restricted? Look at lane number 1. It says M. M is for marker. Okay? It's a molecular weight marker. Basically, it's telling you it's DNA of known size. This is not DNA we're interested in restricting. This is just to tell us what is the size of the other DNA that we have run in the gel. It is a marker. Okay, and this is what you call a 10 kb. It looks like a ladder. Okay, 10 kb. That means there's one. This is 1 kb because it's a small piece. This will be what? 2 kb, 3 kb, 4, 5, 6, 7, 8, 9, 10. Okay, so this is a 10 kb ladder, and all it is there is to tell us what size are these DNAs here that we have restricted. So now that's the M line. Here it says none. This is DNA which we did not digest with the restriction enzyme. We didn't restrict it. Okay? This is just the DNA where we didn't put an enzyme in. This is what unrestricted DNA looks like. A very good DNA extraction will look like this, where you have all the pieces of DNA in what we call high molecular weight. Okay? That means they are all big pieces of DNA. Okay? You don't see how many, you see some at the bigger going to medium, but here you don't see anything. No? That's because there's been no restriction. It's just extracted isolated DNA. Now look at this. This 4 lanes here is this DNA put into 4 other tubes, and in each tube it was restricted by a different restriction enzyme. Here's HIND3, XBA1, EchoR1, BAMH1. Okay, these are known restriction enzymes. You know these are restriction enzymes. What has this BAMH1 done to this DNA? It doesn't look the same anymore. Yes, there are some big pieces remaining, but most of the pieces are here. You see, it's turned this big pieces into millions of different size pieces. So you have big, you have medium, and you have small. Okay? So that is what restricted DNA looks like. Okay? The genomic DNA is a single large band, that one there, unrestricted. But when you restrict, you get this restriction smear, because you have so many millions of fragments, all of different size. Because what? The restriction enzyme was just chopping that DNA in that tube. Okay? So this is the first thing we need to do with our DNA. We need to restrict it. We need to take it from there. We extract, that's the genomic DNA. You can take it. If I spit into a tube, you can get my DNA. Yeah? And run it on this gel here. It will look like this. Then what do? We chop it with restriction enzymes, and they create this profile of restriction enzyme. We are only interested in the small pieces here. We're not interested in these big and medium pieces. We throw them all away, actually. We cut out the DNA from down the bottom here where the pieces are small. Those are the only pieces we're interested in. Okay? So, let's go through the method of RAD-seq. First, you collect the samples and extract the DNA. Then, as we said, you restrict the DNA for each sample, right? Then, you ligate and adapt. Here is the genomic DNA here, right? You see here these restriction enzymes. They're restricting the DNA. They're cutting it at that place. And what do? So they've cut it here, for example. You see my mouse? They've cut it here, and it's resulted in this blue piece here. Then what do? We ligate. Ligate means what? Join, right? DNA ligase is a joiner. It glues things together. It's the opposite of restriction. Restriction cuts. Ligase joins. So we use ligase to join this Illumina adapter here to this piece of restricted DNA. All right? Here's the restriction site where it was cut. That is the orange part. We also add a barcode. Okay? We add an individual barcode. Remember, if I said you want to look at different individuals and sequence them all at the same time, well, this is how you do it. You can do it for Amplicon, but you can also do it here for RAD-seq. You give them different barcodes, and you can run it all at once in the same lane of the NGS machine. All right? You give them a barcode, so this is a unique piece of DNA sequence according to each individual. And then you have to have this Illumina adapter here, because this Illumina adapter is very important. It's this pink one here, and that is the PCR primer. Because once this here goes for NGS, you need a PCR primer on this side and that side to amplify that DNA. To amplify the DNA so that we can sequence it using NGS. Okay, so then what do? We after we've ligated these different things onto these restriction fragments, we pool the samples. That means we put them all together into one tube. Remember, we had different individuals. We restricted each one. We gave each one a different barcode plus the Illumina adapter. Now what do? We put them all together in one tube, right? And then what do? We sequence them. Okay, we can sequence them using single end sequencing or paired end sequencing. We won't get into the details of those in 3649. You will get to that in honors. But then, what do we need to do? We need to shear the DNA. Okay, before we put this second PCR adapter on here, we need to shear the DNA. This is a peculiar thing for RAD-seq. Why do we shear? What is shear? You know shears. When you are shearing a tree in your garden, you're just using those big things. They look like big scissors. They trim the tree. So shear is basically trim the DNA. So basically, you take you don't use restriction enzymes. You use something that vibrates the DNA very, very, very fast, high frequency, and it breaks the DNA into small pieces. Okay, we need small pieces because that is the DNA that we need to sequence using the Illumina technology. Okay, the NGS technology only takes small pieces, so that's why we need to shear the DNA. Then you select the size you want, between 70 and 90 for Illumina sequencing, because as I said, that's the size of piece that gets sequenced in the Illumina reaction. Just it's sequencing millions of little pieces like this, you see. And so after all of their sequence, you have to put them together and assemble the sequence. But actually, the size of the DNA sequences using NGS, at least the second generation of NGS, is between 70 and 90 base pairs. So that's why we shear the DNA and we select only the small pieces, because bigger pieces can't get sequenced. Okay, with that Illumina technology. Okay, then we ligate the other PCR, the P2 adapter, and then we send them for either single end and paired end sequencing. And then what happens is that they get amplified by these two adapters here, and the NGS happens from either single end one side or from both sides, and then we map and call the SNPs. Okay, we map all the SNPs, we line them all up. We line all these millions of pieces. Here you see all the little pieces that have been sequenced, little 70 to 90 base pair sequences, and we need to line them all up like this. Okay, we need to stack them all up and line them up. And so by doing that, we then know the DNA sequence, and the alignment. That's very important, right? Because without having them in a line, you can't say anything about the DNA sequence. You can't say what is the mutation rate. You can't say how much variation there is. You can't say how they are related to each other. Okay? You have to have them in a line, and that's what this mapping is doing. It's mapping them all into lines, the way we would normally do for Sanger sequence for one locus. But this is being done for millions of loci at the same time. Okay, so that's RAD-seq. We'll go through it one more time because it is a little bit confusing. And this time we use two samples. We call the sample 1 S1 and sample 2 S2, right? So let's go through that process again. Now we put on barcodes. We then and the P1 adapter on the one end, okay? And then we pool the samples, put them all together. So now you see the purple and the orange. So we gave the orange barcode to S1, the purple barcode to S2, but now you see we've pulled them together. Do you see that? They are purple and orange ones, both in the same tube. We've put them together. Now what do? We shear. Now look at the size here. We shear them, we break them down, and then we choose only what? The very smallest ones. Then we give them the P2 adapter, we send them for PCR and NGS, and then Illumina sequences them all. Okay? So the barcodes that we put here are always unique to the sample. So that when we put them together, we sequence them. At the end, you see, we are able to separate the sequences based on the barcode. So we know all the ones with the orange that were sequenced belong to sample 1. And all the ones with purple that were sequenced belong to sample 2. Okay? So that's RAD-seq. It has a lot of advantages, okay? You don't need to know anything about the DNA sequence. You don't need to know anything about the DNA sequence, okay? You can do this method on any organism, okay? From as long as you can extract the DNA. You don't need to know anything about, unlike PCR and Sanger, where you need to know a piece for the primers, you need to know the sequence of the primers. For RAD-seq, you don't need to know, All right, you just chop randomly with the restriction enzyme, and you don't need to know anything about that DNA sequences beforehand. In the lab, it's relatively straightforward, and the great advantage is it gets a huge amount of data. Not as much as whole genomes, but still much more than normal Sanger could ever produce. Much more than microsatellites could ever produce. Okay, what are the disadvantages? Every good thing comes with a problem, right? We all know this. Nothing comes for free. So what is the problem with the RAD-seq? Yes, we don't know where the mutations are in the genome. They might be in a gene. They might be under selection. They may not be neutral. This will become more understandable when we continue in this module. You get to the neutral theory. Okay, so we don't know that there's no natural selection working on any of these mutants. Okay, and that's important to know. That's important to know. Another disadvantage is that the number of loci in common to all animals will reduce with the number of samples. So basically, you are randomly cutting the DNA. Okay, you are hoping that you will sequence at random the same pieces of DNA for all the individuals. Okay, so the more individuals you have, the less chance of sequencing as much DNA as possible for all the individuals. All right, that's a big disadvantage. Now we go to genotype by sequencing, which is a method that's very, very similar to RAD sequencing. It's also reduced genome representation, and it has these 4 steps, okay? Except what? Who can see where these steps are not the same as RAD-seq? Huh? Let's go through it. Step 1, here's your genomic DNA. Here are the cut sites for the restriction enzyme. Here you see that the genomic DNA has been digested with this restriction enzyme APEK1, and then it's resulted in these different fragments here. And here's the restriction cut site at the end. What do? We take the DNA sequence, we put on our barcode, we put on our adapter, and then we put on the reverse adapter. Already you are thinking, hang on a sec. In RAD-seq, we did that, but we waited. We didn't put the reverse adapter on immediately. Now the clever ones of you have already figured this out. Then what do? We pull the DNA, okay, and then we simply send it for PCR amplification. And then for Illumina sequencing, it does the sequences are then separated based on the barcode you see here, okay, and you have all your DNA sequences. But what didn't we do that we did do in RAD-seq? Huh? After we pulled in RAD-seq, we did what? We sheared the DNA, remember? We broke it into small pieces. And then we only selected the very smallest ones, right? We don't do that here in GBS. There's no shearing step. The shearing step would come here where the red arrow is. I've put the red arrow there for you. Instead, we restrict, we add the barcode, we add the forward primer, then we also immediately add the reverse primer. And we don't say this has to be a small size. So that means we will add a reverse primer even to a big sized restriction fragment. Okay? And so what do you think, leaving out this shearing step, what do you think it does compared to RAD-seq? Let's see, it will be a disadvantage. We'll go through the advantages first, but think about, what do you think, leaving out that shearing step where you select for the smallest in RAD-seq? We don't do that in GBS. What do you think, is that going to be good for GBS or bad for GBS? What do you think, and why? So let's go through the advantages. It has all the advantages of RAD-seq, right? It can be used from any organism. You don't need to know the sequences, the sequence beforehand, or any primer or anything like that beforehand. It's pretty straightforward in the lab, and you also get a huge amount of data. What are the disadvantages? Same as RAD-seq, you don't know that you're getting selectively neutral polymorphisms, and here we have it. The number of loci, the number of SNPs that you will be able to sequence is less than RAD-seq. Why? Because you didn't do the shearing step to bring them all into a small size that the NGS Illumina machine likes to sequence. Okay? It doesn't like to sequence big pieces. So if you did not shear, and you just put the second adapter, that means there will be some small pieces which will be sequenced, but there will also be some big pieces which will not be sequenced. Okay? And so that's why you end up getting more sequence from RAD-seq, because you did that shearing step. Okay? And that's what I've just explained here. Because without shearing, some fragments will be too big for the Illumina to sequence. It needs this fragments in this size here. Okay? So the shearing step in RAD-seq is very important. I will be asking you about the difference between GBS and RAD-seq. You guys better know it. I will see you in the next lecture. Okay?